Hello students, in this presentation we discuss about the cell structure. As we know, cell structure of the plant and animals, we have already known that in your previous classes. What is the structure of the cell? Now, in the second year, semester 4, paper 4, cell biology, genetics and plant physiology. In this first unit, you have plant cell envelopes and cell organelles, nucleus, chromosome, cell division. In the plant cell envelopes, mainly ultrastructure of cell wall, models of membrane structure, structure and function of the semi-permeable plasma membrane. In cell organelles, mainly we will discuss about the nature of the mitochondria and chloroplast, semi-autonomous nature of mitochondria. And in nucleus subunit, ultrastructure, types and function of the DNA and RNA, mitochondrial DNA, plastids, DNA and plasmids. In chromosomes, morphology, organization of DNA in chromosomes, euchromatin and heterochromatin, karyotype, special type of chromosomes, Lamprecht and Palatin chromosomes. In this unit, most of the in your examination, long bits are come from mainly in the plasma membrane and nucleus, structure of the nucleus and function of the DNA RNA, difference between DNA RNA and chromosomal structure, special type of chromosomes and cell division. So today we will discuss about the plant cell and animal cell. What is the structure of the cell? A cell is considered to be the structural and functional unit of all organisms. Tiny organisms like bacteria, fungi, higher plants and animals, including the most involved in types, are basically made up of independent units called cells. The study of cell is called cytology. Within the availability of various cytological techniques and methods, most sophisticated tools, the intricate details of number of biological phenomena occurring inside the cells came to be understood. In the recent times, the study of cell deals with a broad range of aspects, including the structural, functional and hereditary characters of cells. Hence, in the modern context, the study of cells not only deals with cell structure and types, but also concern various cell organelles and their functions, the inheritance of characters, etc. Therefore, the study of cell in a broad perspective is referred to as cell biology. Okay. The studies of cells evolved into a branch of science within the invention of microscope by Janssen. According to Robert Hooke, discovered plant cells while observing thin slice of bark in microscope. Hooke described his observation in the volume Micrographia. Later in 1674, Anton van Leeuwenhoek described the structure of red blood cells, protozoa and bacteria. The term protoplasm coined by Purkinje. Term protoplasm was Purkinje. Robert Brown in 1831 earlier proposed the presence of spherical ball-like structure called nucleus. This is the nucleus. So term Robert Brown earlier reported the presence of spherical ball-like structure is called the nucleus in the protoplasm of cells. The beginning of the 19th century contributed a major information about the plant and animal cells. Cytological research progressed in view of the availability of modern methods, tools, techniques and advanced microscope technology. Okay, German botanist M.J. Schleiden in 1838 and geologist T. Schwann in 1839 proposed the cell theory. According to the cell theory, all living organisms are made up of cells. Cell is the structural and functional unit of all organisms. Rudolf Virchow in 1855 in his famous aphorism Hominis cellula e cellula, that's mean discovered that new cells arise from pre-existing cells. Within this discovery, one more aspect was added to the cell theory, that the cell is hereditary unit of all organisms. Then cells are of two types, that is the prokaryotic cells and eukaryotic cells. In prokaryotic cells, these are the found in primitive organisms such as bacteria and blue-green algae. Blue-green algae also called the cyanobacteria. In these cells possess a distinct cell wall and protoplasm, but well-defined nucleus with a nuclear membrane is absent. The nucleus is presented by a single circular naked DNA molecule, which is generally referred to as a nucleide. You can see in the diagram, this is the bacterial cell. In this nucleide, the nucleus is represented by a single circular DNA molecule, which is referred to as a nucleide. In this cell, absent in the nuclear membrane. Okay, this is the eukaryotic cell. This one is the prokaryotic cell. Okay, in this eukaryotic cells, these are found in all major plants and animal groups. These cells are considered more advanced and have a complex cell organization. These possess a well-defined and definite nucleus with a nuclear membrane, chromatin and nucleus. We can see in this diagram, this is the eukaryotic cells. In this eukaryotic cells, this is the nucleus. In this nucleus, they have nucleolus, nuclear membrane. This is the nuclear membrane, and chromatin and nuclear sap. This is the chromatin, the light brown is the chromatin. Hair-like structures are present in this, and this is the nucleus sac. This is the eukaryotic cell, three-dimensional structure. We can see in the center of the, this is the nucleus, this is the nucleolus, this one is the mitochondria, this one is the endoplasmic reticulum. This is the Golgi bodies, and this outer layer is the plant cell wall, and this one is the inner layer is the plasma lemma, that is the cell membrane, and this one is the centrioles. Okay, then what is the difference between plant cell and animal cell? Coming to the animal cell and plant cell, we discuss what is the difference between the animal cell and plant cell. In this animal cell, this is the animal cell, this one is the plant cell. In this animal cell, they are the generally smaller in size in animal cells, but in the plant cell they are usually larger than animal cells. In this cell wall is, in this animal cell wall is absent, but in this cell wall is present in addition to plasma membrane and consists of medulla, primary and secondary wall. Okay, in the animal cell, vacuoles are small in size, but in the plant cell, vacuoles are larger in size. We can see in this animal cell, these are the vacuoles. But in these vacuoles, small and temporary. But in the plant cell, vacuoles are larger in size but permanent. Okay, this is the vacuole. This one is the big size like structure. Then in this animal cell, tonoplast absent. In this plant cell, this is the vacuole. In the vacuole, surrounding layer is called the tonoplast. This tonoplast is present in the plant cell, but in the animal cell, absent. Centrioles present in the animal cells, but in the plant cell, centrioles absent, except motile cells of lower plants. Okay. Then nucleus at the center of the cell. Nucleus at the center of the cell in animal cell present. We can see this is the nucleus. This is the animal cell structure. In the center of the cell, nucleus is present. But in the nucleus present along the periphery of the cell. In the plant cell, in nucleus is the in the side of the in the cell structure. You can see nucleus present along the periphery of the cell. In this animal cell, lysosomes present, but in the lysosomes are rare. Where plant cells, lysosomes are rare. In the animal cell, storage material is glycogen granules, but in the plant cell, storage material is starch grains. Okay, this is the what difference between the animal cell and plant cell. In this, this is the cell wall. Cell wall is the ridge and storage wall. Protects, maintenance the shape of the cell. So, we will discuss later about this cell wall in detail. Okay, this is the brief structure. We can see in the cell, outer layer is the, this is the cell wall. This is the rigid and strong wall. This is the present in the only plants. Okay, then in the plant cell membrane, every cell is enclosed by a cell membrane. It controls the passage of material in and out of the cell. This is also we discuss later in detail. This is the long bit, very important question, cell membrane. Okay, today we only discuss about the briefly about the what is the cell structure, what is the organelles are present. Okay, then nucleus. What is the nucleus? This is also very important bit. We discuss about the in detail what is the nucleus, what is how many types are uh, present, what is the cell membrane, nucleus membrane. So the control center of the cell, it contains the DNA code for cell coiled into chromosomes. Okay, this is the nucleus. We can see in the nucleus, nucleolus present. Okay, and chromosomes, chromatids, nuclear pores, all are present. Main function of the nucleus, we can see this is the nucleolus. This one is the chromosomes. This is the nuclear envelope. Nuclear envelope absent in prokaryotic cells. In nuclear pores, we can see in the nuclear pores also present. This is the chromatin. Okay, function of the nucleus, it is control and regulate the function of all the cell organelles. It is therefore referred to as a dynamic center of cell or cell brain. It plays an important role in heredity. This is the nucleus. Then next we will go for the endoplasm reticulum. In the endoplasm reticulum, there are the two types of endoplasm reticulums present. What are they? It is a network of lipoprotein microtubules extending from the outer nuclear membrane to the plasma membrane. We can see this is the nucleus. In the nucleus, outer nuclear membrane and to the plasma membrane. In the lipoprotein microtubules like structure extending from the outer nuclear membrane to the plasma membrane. So it was reported by K.R. Porter in 1945. It is found in all eukaryotic cells. It is composed of cisternae, vesicles and tubules. Three types of structures are present. In this mainly granular ER and agranular ER and are rough endoplasmic reticulum, smooth endoplasmic reticulum. So in the rough endoplasm reticulum, this is also granular endoplasm reticulum. In this type, the surface of membrane is studded with a ribosomes and has rough structure. You can see this is the endoplasm structure. In this structure, in this type of the membrane is studded with the ribosomes, attached to the ribosomes. In this we can see in this layers tube-like structures, lipoprotein microtubules like structures. These are attached to the ribosomes. So it is rough structure. In agranular ER, these are the agranular ER. These are the also called the smooth endoplasmic reticulum. This is also called the smooth endoplasmic reticulum. In this type, the ER membranes do not show ribosomes and appear with smooth surface. So mainly, what is the function of the endoplasm reticulum? It takes part in protein and lipid synthesis. Rough ER, that is the rough endoplasm reticulum, is concerned with protein synthesis. Smooth ER is concerned with lipid synthesis. Okay. Then we go for the ribosomes. This is not an organelles but very important. In these ribosomes, these are tiny structures were first proposed by the Ballade in 1955 with electron microscopic observations. They are also known as the ergosomes. They are the ribonucleoprotein granules and present in all kinds of cells. They are produced in the nucleus and later move into cytoplasm, where they appear as free floating masses or attached to the surface of endoplasm reticulum membranes. Okay, this is the free floating like structures are present and also attached to the endoplasm reticulum. So, in eukaryotic cells, UATS and 70S type of ribosomes are present, and in prokaryotic cells, only 70S type of ribosomes are present or found. This is the only prokaryotic cells, only 70S type of ribosomes are found, but in the eukaryotic cells, ATS and 70S, both types of ribosomes are present in eukaryotic cells. The classification of the ribosomes as ATS and 70S based on the rate of sedimentation coefficient. This is the unit of measures. S means Swedberg unit. S means Swedberg. S means Swedberg unit. This is the sedimentation coefficient during the ultracentrifugation process. Okay. So they are the composed of two unequal subunits. You can see in the diagram also. This is the smaller unit. This one is the bigger unit in the 70S. And also in 80S, this is the smaller unit. This one is the bigger unit. This one is the bigger unit. In the 70S, ribosomes are composed of 50S and 30S subunits, while in 80S, ribosomes show 60S and 40S. This one is the 60S, this one is the 40S subunits are present. The subunits of ribosomes are held together with help of magnesium ion. This is the main important. These both subunits of ribosomes are held together with the help of magnesium ion. This is the main important. This is the very completed bit. Magnesium ion. During the protein synthesis, several ribosomes form into long chains called polysomes or polyribosomes. So, during the protein synthesis, several ribosomes form into long chain-like structures that is called polysomes or polyribosomes. Okay. Main function of the ribosomes, they are involved in protein synthesis. Okay. This is the ribosome structure. Then Golgi complex. What about the Golgi complex? The Golgi complex, it was first reported by Golgi in 1898. It occurs in eukaryotic cells, appears as a group of membrane bound structures. They are also termed as lipochondria. What? Lipochondria or idiosomes. In plant cells, about 1 to 15 Golgi units are found, and these are known as dictyosome, Each dictyosome is comprised of 3 to 7 flattened sacs called cisternae. 3 to 7 flattened sacs called cisternae. These are the cisternae. See in the diagram also. Okay. Few spherical large vacuoles and tubules which are bounded by single unit membrane and filled by an amorphous substances. In this Golgi complex, main function of the Golgi complex perform the following some functions are present. That is the synthesis of cell wall materials like cellulose, hemicellulose, pectin and polysaccharides. Another function, secretion of lipids, hormones and enzymes, and production of lysosomes, formation of cell plate during the cell division. This is the main function of the Golgi complex. Then lysosome. This is the lysosomes. This one is the lysosome. Main breaks down material for digestion, contains special enzymes for digestion in the cell. They are spherical single membrane bounded organelles filled with enzymatic matrix which is rich in hydrolytic and phospholytic enzymes. They are found in large number in actively dividing meristematic cells. They were first reported by Christian D. Dubé. They originated from endoplasm reticulum or Golgi complex. Lysosomes can be identified into three types: primary lysosomes, secondary lysosomes, tertiary lysosomes. Okay. In this primary lysosomes, these are the spherical vesicles filled with enzymatic fluid, and the secondary lysosome, these are spherical vesicles which show the presence of food molecules or microorganisms. In the secondary lysosomes, presence of food molecules and microorganisms. In tertiary lysosome, these are spherical vesicles which contain undigested food materials. These are also called residual bodies. The tertiary lysosomes, these are the spherical vessels which contain undigested food material. These are also called the residual bodies. This is the central vacuole. What is that? We already discussed about what is the difference between the animal and plant cell. In that there are the vacuoles small and temporary in the animal cell, but in the vacuole large and permanent in the plant cells. Yes, this is the central vacuole present only in the plant cell only. Most plant cells have one large one and filled with fluid and helps maintain structure, pressure and shape of the cells. In this the central vacuole, you can see this is okay. Then next we will study, we will discuss about the mitochondria and chloroplast. This is the main in your cell bus. You can see 1 minute. The first in your cell bus, plant cell envelopes, that is the mainly cell wall and membrane structure, function of the semi-permeable plasma membrane. The cell organelles, we already briefly what is the structure of the cell, and then we will in detail discuss about the mitochondria and chloroplast. Okay, thank you. Tomorrow we will discuss about this mitochondria and chloroplast. Thank you.